Hello and welcome to IB Biology Notes with Dr. Kimmo. This is A2.2 Cell Structure. This chapter is about cells and microscopy, and you might want to press pause to have a look at the contents. The cell is the fundamental unit of life. The term cell was coined by the Englishman Robert Hooke, who first observed cells in cork. He made the drawing on the right, and he didn't really understand what cells were and how they worked back then. But as scientists studied more and more organisms, cells were observed in all of them. It was concluded that all living things consist of cells. This is an example of a theory based on observation. A general prediction was made, although not all organisms and tissues had been studied. Modern cell theory is a more developed version of this original statement, and more about that later on. Today we know of some organisms with atypical cells, but the theory still holds. It has not been falsified. The light microscope is a very important tool for biologists. And you need a light microscope to see a cell. Most modern microscopes are compound microscopes with two or more sets of magnifying lenses, one in the eyepiece and one in the objective. Now, an objective might contain several lenses, but it's still one set of lenses. For example, if you have a 10x eyepiece, that's an eyepiece that has a 10 times magnification, and you have a 40x objective, then you have a magnification of 400x. 10 times 40 is 400. The microscope illustrated here has 4 objectives attached to an objective turret, also known as a revolving nosepiece. Now let's have a look at the different parts of a microscope. Starting from the top, the eyepiece, you might have 1 or 2 of them. If you have 2, it's important that you adjust them so that they fit the distance between your pupils. Then you have the turret. This one has 4 objectives on it. Under the objectives, there's the stage. This is where you put your specimen. And in a microscope like this, the specimen is always on a microscope slide and has a cover slip on top. The condenser under the stage, it directs the light so that the specimen will have optimal illumination. Further below, somewhere here, you have a light intensity knob and you have the light source. So you have a lamp underneath inside the base. On the right side, from top to bottom, you have the stand, and the stand and the base at the bottom, those are the parts that you should grab when you move a microscope. So don't grab the eyepiece, don't grab the objective turret, the stage, anything else, just the stand and the base. Here you also have a stage clamp, which keeps your specimen in place. Below you have the focus knobs, coarse focus, that you use first, and then you fine tune with the fine focus. And finally, you have the stage control knobs, left, right, and forward, back. And it might take some time to get used to this, but it's very practical once you get it. Magnification. Let's say you look through a school microscope with a 10x eyepiece and a 40x objective. Then you are magnifying the specimen by a factor of 400. Everything looks 400 times as big as it would look without a microscope. If you take a picture of what you see, the magnification will depend on the size of the picture. So the size of the object in the picture divided by the actual size of the object. Let's say that the diameter of this circle on my screen is 90 millimeters. It might be different on your screen. On my screen, it's 90 millimeters. Let's say that I know that the field of view through this microscope is always 0.18 millimeters. 90 millimeters divided by 0.18 millimeters is 500. That means that the picture on my screen right now has magnification of 500x. Now, this is not always practical because I don't know if you are watching this video on your phone or on a big screen. So I can tell you that the magnification of this picture is 500x on your screen. That's why it's very common to add a scale bar to a micrograph. The scale bar stays the same relative to the micrograph. So even if you see the same thing in smaller size, the scale bar follows the shrinking, and you can always tell more or less how big the objects in the picture are. Let's look at some examples on how to calculate specimen size and image magnification from micrographs. On the left, you have a micrograph with a scale bar, and let's say you want to find out the size of this stoma on a leaf. And let's say it's printed on a paper, maybe an exam paper. So where do we start? Well, first you take your ruler and you measure the specimen. And let's say it looks like it's 61 millimeters long on the paper. Then you measure the scale bar, which seems to be 18 millimeters. Then you calculate the specimen to scale bar ratio. So 61 divided by 18, that's 3.4. And finally, you multiply the scale bar marking, 5 micrometers, with the ratio that you calculated. 5 times 3.4 is 17. So the length of this specimen of the stoma is 17 micrometers. All right, let's say you want to find out the image magnification based on the scale bar. So first you measure the scale bar. It looks like it's 7 millimeters. Then you convert that number to the same unit as the scale bar marking. The scale bar marking uses micrometers. So the length of the scale bar is 7,000 micrometers on your paper. Finally, you divide the scale bar length that you measured with the marking. So 7,000 micrometers divided by 5 micrometers, that's 1,400. That is the magnification of this paper, of this micrograph. And finally, on the right, let's say you want to find out the specimen size based on magnification. Now you can see it's the same picture as in the middle. It's also a stoma, seen from the side. And we know already that the magnification is 1,400. All right, so there you measure the specimen. In this example, let's say you want to find out the length of one of these cells, this one. You measure it, it looks like it's 39 millimeters. You convert that to micrometers, 39,000 micrometers, and then specimen divided by magnification. 39,000 micrometers divided by 1,400. That's 26 micrometers. That is the length of the cell. The first microscopes of the 17th century did not produce the clear images that we're used to seeing today. The main challenge was to shape the lenses correctly, so there was a lot of trial and error. On the left, one of the pioneers, Robert Hooke, although historians can't agree on whether it's really him in the picture. Anyway, the microscope on the left, that's one of the microscopes he used, and he used compound microscopes. On the right, another pioneer, Antony van Leeuwenhoek, a replica of a single lens microscope that he built and used. So only one lens, but a very powerful one. Increased resolution and image quality. With the development of optical theory and improved techniques in the 19th century, microscopes became better and better. Optical theory meant that manufacturers could calculate the optimal size and curvature of the lenses and the distances between lenses and things like that. And the Industrial Revolution brought with it a lot of improved techniques for manufacturing microscopes and other things. The first mass-produced microscopes were made by Carl Zeiss in Germany in the middle of the 19th century. The maximum theoretical resolution of the light microscope is approximately 0.2 micrometers, and this is a physical constraint that has to do with the wavelength of visible light. However, some modern laser-based instruments can achieve higher resolution, for example STED microscopes, 0.05 micrometers or less. Their use is, however, much more limited than a normal, regular light microscope. Then there are electron microscopes, which use an electron beam instead of light, and they have a maximum resolution of 0.001 micrometers, so that's 1 nanometer. Fluorescence microscopy. Most tissues appear transparent under the microscope and cannot be seen unless they are stained or dyed. You may have used some regular dyes in the lab and then looked at your specimen through the microscope and you can see different structures in different colors. Fluorescent dyes are also very popular. Not so much in schools because you need a special microscope to be able to see them, but very common at universities, in life science companies. Fluorescence is when a molecule absorbs light and emits it back at another slightly longer wavelength. For example, glow-in-the-dark stars that you can attach to your ceiling, that's fluorescence. If a cell is stained with a green fluorescent dye, that dye cannot be seen until it's exposed to a beam of blue light, which causes it to appear green against a dark background. So first you see nothing, then when you turn on the blue light, you see the green fluorescent dye. The example at the top is of a blue fluorescent dye. The cell nuclei in the hippocampus region of a rat brain emit blue light when exposed to UV light. It's because the specimen has been stained with a blue fluorescent dye. This dye binds to DNA. So when you turn on the UV lamp on the microscope, the DNA in the nuclei of the cells appear blue. So each dot is a cell nucleus in this picture. On the right, another example, a section of a neuronal dendrite in a thin slice of rat brain cortex. The fluorescent dye was injected into a single neuron, and the black background is packed with dendrites from other neurons, but these cannot be seen because they have not been stained. This way you can study this single dendrite and see all the details like the small spines, which are where synapses are located. This is where the neuron makes contact with other cells, mostly neurons. Immunofluorescence. Fluorescence microscopy is often combined with immunofluorescence, IF. IF involves antibodies which carry fluorescent molecules. And you may remember antibodies from immunology. Those are the molecules that recognize viruses and bacteria, other pathogens that may infect you. Most immunofluorescence methods involve two sets of antibodies. Primary antibodies, which bind exclusively to the antigen, for example, protein, that you want to be able to see under the microscope. So in your body, antibodies are produced to recognize pathogens like viruses and bacteria, but you can actually also produce antibodies against a lot of other things. For example, proteins that you want to study. Here, the primary antibody binds to the antigen, for example, protein. Then you add secondary antibodies, which bind to the primary antibodies. These secondary antibodies are carrying fluorescent molecules. Later, when you look at your specimen through the microscope, wherever the antigen is located, you see green light in this example. It could be another color. Here it's green. Several fluorescent dyes can be used together. This panel shows an overview of the cotton rat from an article by Rashidul Islam and others. Here, follicles were detected with an antibody against a follicle protein called DDX4. This antibody would be a primary antibody, so it binds exclusively to DDX4. Then a secondary antibody was added, and this secondary antibody was carrying green fluorescent molecules. The secondary antibody recognized the primary antibody, and now we see DDX4 in green. Epithelial cells were detected with an antibody against an epithelial protein called FOXL2. This antibody would also be a so-called primary antibody, and afterwards a secondary antibody that detects this primary antibody was added, and the secondary antibody was also carrying red fluorescent molecules. So now we see FOXL2 in red. Finally, cell nuclei were dyed with a DNA marker called Hoechst. This is not immunofluorescence. It does not involve antibodies. It's just a molecule called Hoechst which binds to DNA. And this molecule is fluorescent. It's blue. Then you can study all these colors separately and take pictures, put the pictures together to get a multicolored image. Electron microscopy can be used for structures that are too small to be seen with a regular light microscope. Here, beams of electrons are used instead of visible light. There are two main types: transmission electron microscopy, TEM, and scanning electron microscopy, SEM. In TEM, electrons move through the specimen, much like light moves through a thin specimen viewed in a school microscope. That would be the picture at the top. In SEM, electrons bump off the surface of the specimen, creating a 3D effect. That would be the picture at the bottom. It's a bit like using a flashlight to shine light from the side. Freeze fracture can be used in electron microscopy. It is a technique that cracks open frozen specimens to reveal their inner structures, and these can then be seen with SEM. So first you freeze and dry the specimen, then you crack it open. Now we move from microscopy to cells. First, what do all cells have? There are three things here. All cells have a plasma membrane or a cell membrane. That's the outer boundary of the cell, and it regulates what moves in and out. It allows the cell to regulate its internal conditions. The concentrations of different substances are often very different inside versus outside, thanks to the membrane. And the general structure of the membrane is two layers of lipid molecules. There's more to it, that's for later. DNA. DNA contains all the instructions a cell needs to survive, especially how to assemble all the thousands of proteins that act as enzymes or perform other specialized functions. Thirdly, cytoplasm, a watery substance that fills the interior of the cell. Many substances are dissolved or suspended in the cytoplasm, and many metabolic reactions take place here. Proteins and other biomolecules that the cell needs to carry out its functions are produced here. Let's have a look now at the simplest cells, prokaryotes, always unicellular. The bacteria and the archaea are prokaryotes, and they have one circular and naked DNA molecule. Naked means not associated with protein. They also have a nucleoid. It's not a real nucleus. It doesn't have a membrane around it. It's more like a region where the DNA is located. They have a cell wall like fungi and plants, but it's made of peptidoglycan, so it's not the same as fungi and plants. They often have a capsule around the cell wall, and they have plasmids, tiny circular DNA molecules that can be exchanged between prokaryotes. They have 70 S ribosomes. The point here is that their ribosomes are different from those in eukaryotes. They have pili, which are used for different things, for example, to attach to other cells. And they have a flagellum, which allows them to move. Eukaryotes, more complicated. This is an animal cell. It's compartmentalized. It has membrane-bound organelles, for example, Golgi apparatus, lysosomes, endoplasmic reticulum. These compartments have specialized functions. Eukaryotes also have a nucleus with DNA in chromosomes, several chromosomes inside the nucleus. Eukaryotes have mitochondria. They carry out aerobic respiration. That's respiration using oxygen. Eukaryotes have 80 S ribosomes to make protein. And again, the point here is that it's different from the ribosomes in the prokaryotes. Plant cells are also eukaryotes. And there are a few things that are different between plant and animal cells. So, cell wall. Animal cells don't have that. Central vacuole for structural support and food storage. And this vacuole is permanent. It's always there. And chloroplasts. That's where photosynthesis takes place. In multicellular organisms, like animals, there are many types of specialized cells, and they all do different things. But in unicellular organisms, like Chlamydomonas here, one cell is responsible for all life processes, such as homeostasis, it's like balance, maintaining internal conditions, metabolism, all the biochemical reactions of life, nutrition for energy and growth, whether it's making your own food through photosynthesis or consuming other organisms. Now, Chlamydomonas can actually do both. Excretion, getting rid of waste, growth, an increase in size or number of cells, movement or transport. So even if some organisms cannot move around, like trees, they can still move stuff around. Response is being able to react to stimuli. And reproduction, production of offspring. This is another example of a unicellular multi-talented cell. Paramecium, it's a unicellular ciliate, and maybe you've already seen some under the microscope. Differences in cell structure between animals, fungi, and plants. First, cell wall. Animals don't have that. Fungi and plants do, but they are not made of the same thing. Plastids. Animals and fungi don't have that. Plants do. They have chloroplasts and amyloplasts. Chloroplasts, that's where chlorophyll is located, that's where photosynthesis happens. And amyloplasts, that's where plants store starch. Related to plastids, food strategy. Animals are heterotrophs and fungi are heterotrophs. But animals have internal digestion. We have a digestive system, whereas fungi, they have external digestion. They don't have a digestive system inside them. Instead, they release enzymes and break down their food externally, outside, and then they absorb the nutrients afterwards. Plants, they are not heterotrophs, they are autotrophs. They produce their own food through photosynthesis. The role in the food web: animals are consumers, fungi are decomposers, and plants are producers. Energy storing polysaccharide. Animals store energy in the form of glycogen. You have glycogen in your muscles, in your liver. Fungi also produce glycogen, but plants produce starch. Vacuole. Animals have small and temporary vacuoles, but fungi and plants have large and permanent vacuoles. Motile cilia and flagella. Now, motile means related to movement. Animals have many cells with motile cilia or flagella. For example, male gametes, sperm cells, and also a lot of cells in the respiratory tract. For example, there are a lot of cilia that keep moving mucus up the respiratory tract to clean it. Fungi don't have cilia or motile cilia or flagella, except flagella in male gametes that swim, and not all male gametes swim in fungi. Plants, same thing there. No motile cilia or flagella generally, except flagella in male gametes that swim. And again, only in some species do male gametes swim. Centrioles. In animals, all cells have that. Those are what make the spindle fibers during mitosis and meiosis. And there are also centrioles in cilia and flagella. For fungi and plants, they don't have centrioles, except in those few male gametes that swim. You know that eukaryotic cells have a nucleus, but red blood cells don't. They lose it during cell maturation. Skeletal muscle cells are also atypical because they are multinucleate. They have several nuclei. That's because several cells fuse to form long muscle fibers. Some fungal cells undergo mitosis without dividing. It means that they copy their DNA, they copy their whole nucleus, but they don't divide. And this results in long filamentous structures called hyphae with several nuclei. Phloem cells, which transport photosynthesis products in the plant, lose their nucleus and most of the organelles, and they survive only with the help of so-called companion cells. You should be able to recognize some cell types and cell structures in light and electron micrographs. So let's have a look at some of them. Maybe you've stained human cheek cells in the lab already. If you have, you may have recognize the nucleus and the cytoplasm and the membrane. You may have looked at a human blood smear, and there you can recognize red blood cells. They are the smaller type of cells. They don't have a nucleus. And the white blood cells, they are larger. They have a nucleus. And you can see how it's clearly stained in the middle of the cells. These two white blood cells are different types, so they look a bit different. And you may have looked at plant leaf cells. Here you can see the cell wall very clearly and chloroplasts. Structures in TEM images, transmission electron microscope. At the top, mitochondrion. It has an outer membrane and an inner membrane, and the space between is called the intermembrane space. These structures, perpendicular structures that you see, are called cristae. And what's not in the intermembrane space is in the matrix. And do not confuse this with cytoplasm, because the mitochondrion is not a cell, it's an organelle. Rough endoplasmic reticulum, RER, all the dots that you see there are ribosomes. On the right side, you can see a Golgi apparatus within the circle. And also here you can see some ribosomes, and you can see part of the RER. To the left, a nucleus and lysosomes. And the nucleus is surrounded by the nuclear envelope. Inside, there's tightly packed DNA, heterochromatin, and lightly packed DNA, euchromatin. The genes that are expressed are generally in euchromatin, and the genes that are sort of stacked away, not expressed, are in heterochromatin. And there's a lysosome. There are several of them in this micrograph. To the right, chromosomes have aligned at the metaphase plate. And below anaphase, you can see the sister chromatids being pulled towards opposite ends of the cell. You can see the centrosome and the spindle fibers. These types of pictures on the right are what you will be studying when you're learning about mitosis and meiosis. Now we are moving to higher level territory. Endosymbiosis. Endo means inside, and symbiosis is a mutually beneficial relationship between organisms of different species. So kind of a win-win situation. Eukaryotes are believed to have formed from prokaryotes through endosymbiosis. More than 2 billion years ago, aerobic bacteria entered an ancestral eukaryotic cell and stayed inside. Later, cyanobacteria also entered one of these cells and stayed. The aerobic bacteria and the cyanobacteria developed a symbiotic relationship with their host cell. Over time, the aerobic bacteria became mitochondria and the cyanobacteria became chloroplasts. Evidence for endosymbiosis. There are 4 points here. First, mitochondria and chloroplasts have a double membrane, and that's what you would expect from a small cell that has been engulfed by a large cell. So the small cell inside the large one would keep its own membrane and the membrane that was used by the large cell to engulf the small one. 2. Mitochondria and chloroplasts have their own DNA, and this DNA is similar to prokaryotic DNA. 3. They have 70 S ribosomes. So these ribosomes are different from the rest of the ribosomes inside the eukaryotic cell. It also means that mitochondria and chloroplasts are able to produce their own proteins. And 4, these organelles multiply by division. So they cannot be synthesized by the cell like everything else in the cell. The cell can build them. But these organelles, they multiply by division, just like prokaryotes do. Here's a great illustration of endosymbiosis, where you can also see how it was preceded by the cell membrane folding to form the nuclear membrane and the endoplasmic reticulum and other endomembrane components. Multicellular organisms have many different types of cells. They are all highly specialized with a structure and function adapted to their specific role in the body. All these cells develop from stem cells during embryonic development. So soon after fertilization, the stem cells can become anything. And over time they specialize to form, in humans, for example, muscle cells, fat cells, bone cells, blood cells, and everything else. This process of specialization is called differentiation, and it happens step by step. In some cells, further differentiation takes place throughout life. Once a cell has started to differentiate, it cannot take a step back. At the top, you can see a multipotent stem cell that can differentiate into many different cell types. This multipotent stem cell is a hematopoietic stem cell. It means it can differentiate into any type of blood cell, but nothing else, not fat cells, bone cells, nervous cells. These stem cells are found in the red bone marrow of most large bones. Let's look at these steps. First, the hematopoietic stem cell can choose between becoming a myeloid progenitor or lymphoid progenitor. The myeloid progenitor can choose between different alternatives, including erythrocyte, which is red blood cell. And from there on, it might choose instead to become a myeloblast, and the myeloblast might develop into a monocyte, the monocyte finally into a macrophage, which is a type of white blood cell that kind of eats bacteria. Now that's just an example, and this whole graph is an example. You're not expected to know all these fancy names and this exact procedure. We'll finish off this long chapter with the evolution of multicellularity. Multicellularity offers several advantages and has developed several times. We know it's developed several times because there are groups of organisms where some species are unicellular, some are multicellular, and some might even be colonial, which is kind of in between. In a multicellular organism, each cell is helped and or protected by its neighbors. For example, the cells in your body live in a more favorable environment than they would if they were swimming about on their own. And in fact, they have lost the ability to swim about on their own because in most multicellular organisms, cells are highly specialized and are not able to survive alone. Multicellular organisms are also more evolutionarily adaptable. That's all for now. See you next time.